So I was thinking, these guys and gals, when they're not hanging around, sitting around, they fly, and they fly in the air. So just consider this another air talk. Anyway, I want to thank the people that do all the work, and in this case in particular, it's Dr. Craig Hebert, who works for me in Ottawa at the National Wildlife Research Centre. This is his project. He's the PI on it. I'm going to thank the birds as well, and their eggs. That's what I'm going to be talking about in this presentation. So this is Craig on the top left there. These are the birds. So it's 5 species we're dealing with. Those are their eggs directly below, just for so that you know relative sizes and things like that. And then below that, there's 4-letter codes for each of the individual species, and you'll see these codes in some of the slides. So I thought I'd put them on the opening slide here. It's really simple monitoring technique. You go to a colony, a nesting colony, and you collect eggs, and you bring them back, and you analyze them. We've been doing this, our group has been doing this for a long time. And so a paper came out, and this was first authored by Craig Hebert in 1999. And in 1999, him and his co-authors, Craig and his co-authors, summarized a quarter century of using herring gull eggs as a monitoring system for contaminants in the Great Lakes. Another person on that paper was Dr. Chip Weslow, and we'll be coming back to Dr. Chip Weslow in a bit. So it's a big program. It's been going on for a long time. These guys have been engaged in this program for a long time, Craig Hebert and Chip Weslow. In fact, Chip Weslow has been doing this in the Great Lakes for such a long time that there's a colony named after him. So if you go to colony 11, I think it is, that's Weslow Rocks. So again, simple thing, you go to a colony, you go to an island where there's a nesting colony of species, one species at least, but maybe more. You collect eggs and you bring them back. It's because it's been going on for such a long time and it's been used in so many different systems, it's very highly standardized. And so there are SOPs, standard operating procedures, for the egg collection, for what you do with those eggs when you bring them back. For instance, at the National Wildlife Research Centre, we have standard operating procedures just for processing eggs. It's used for international programs. For instance, this monitoring technique with herring gulls at least is used for international monitoring programs like the Great Lakes Action Plan, the IJC, which is the International Joint Commission, which is binational management system for the Great Lakes, and the Northern Contaminants Program is another program that uses these eggs for monitoring purposes. So what happened was Chip Weslow was in western Lake Athabasca in 1977, went to Egg Island and collected some eggs, California gull eggs. He deposited those eggs into the National Wildlife Specimen Bank, which is at the National Wildlife Research Centre where I work, where they were held in our frozen tissue archive for decades at minus 40 in our freezers. And those eggs were eventually pulled in 2009 by Craig Hebert and analyzed for mercury and some other contaminants. And that resulted, because those eggs had been collected in 77 and kept and stored in our tissue bank, we were able to pull those eggs and analyze them. And that gave us our baseline data point for egg contaminant levels in the oil sands region. And this is it. This is our baseline, our data level, our baseline data for egg contaminants from Egg Island in western Lake Athabasca from 1977. So what Craig decided to do was he went back to Egg Island, which is the easternmost yellow star there in western Lake Athabasca, to collect eggs again, and this was in 2009. He also went to another colony, a nesting colony, with the collaboration from Wood Buffalo National Park and Parks Canada people in Mamui Lake, and that's the more southern yellow star, and collected eggs from there. And he also went to another colony that's not on this map, but it's on the Peace River at Rocky Point, And that was a promising colony just because it separated from inputs from the Athabasca River. Now, one of the reasons that these colonies were of such interest to us for collection was because they receive waters from the Athabasca River. And this is just kind of a bad scan of a flow diagram where you can see the Athabasca River coming into the Peace Athabasca Delta and where those colonies are located with respect to that river flow coming in. You can see the colonies too, Egg Island again and Mamwe Lake, where they're nesting on sort of floating debris and stuff like that. So Craig in 2009 did get these eggs, and what he did was he compared them to the 1977 eggs and published a paper basically comparing the levels in those eggs from the more recent collections and from the 1977 eggs. I should mention the 77 eggs because they were analyzed with the more recent eggs, so they're analyzed using exactly the same instruments, the same techniques. So they were comparable. And JOSM came around after that, and with JOSM funding, we decided to continue this sampling program. So Craig, again, has been going to Mamwe Lake and Egg Island, and with JOSM, he added another colony. This is at Langdon Reservoirs near Calgary, and this is basically to establish sort of a northern contaminant picture and a southern contaminant picture for Alberta. Same process, brought eggs back, analyzed them for mercury, for stable isotopes to look at food web structure, and other contaminants. So for example, we've heard some mentions this afternoon about fire. And so one of the things that Craig did was compared, he looked at the dibenzodioxin and the dibenzofuran levels in those eggs to see if there was a signature from the fires, for instance, the Richardson Backcountry Fire of 2011, I think it was, to see if there's any suggestion that the contaminants that he was, in particular mercury, that he was measuring in those eggs, if there's any suggestion that that might have been a result of the big fires that happened before those eggs, a year before those eggs were collected. So this is some of the data. I'm just going to go through it really quickly. And again, is these four-letter codes. So so he's adding through JOSM, he's adding just annual data collections to this program. And so Egg Island tern eggs, Mamwe Lake tern and gull eggs, and then he published a paper. And basically what he did in this paper, he compared the more recent data against the most earliest collected data and looked at all the other relationships that he's seeing. Because he had the data from southern Alberta at Langdon Reservoir near Calgary, he could compare what was going on in the north and what was going on in the south. Interesting information that came out of that was the southern collection sites seemed to be declining over the years in terms of mercury in those eggs over time. Opposite trend going on, or opposite pattern going on with the northern eggs where they were going up. And in the final year, there was a significant difference between the egg mercury concentrations in the eggs from Egg Island from the Calgary site. So these are comparing just California gull eggs that have been collected from those two sites over the years. And so there's the difference in pattern that's occurring between the more northern and the southern eggs suggests that there's a local source for the mercury that is a different source. There's a local source for the mercury in those eggs in the north because they were basically taking a different path from the birds in the south. Through JOSM, going back each year after that and collecting more information, and what we're seeing now after the sort of the high year in 2012 was interannual variability, not surprisingly. So back at Egg Island with the terns, going down in 2013, terns and gulls at Mamwe Lake, again down in 2013. And then finally with the California gulls from Egg Island, and this is the one place with the one species where we have this, what might be considered, I guess, a long-term data set from 1977 up to the most recent collections. So the pattern that we're seeing there with Egg Island and Western Lake Athabasca and the California gulls is that the egg mercury levels have increased in the more recent collection years compared to the earliest year of collection. So that's a fairly interesting result. So what's going on? This is a so what question. Well, one of the things that we're looking at is the fact that there's information on embryo toxicity, or there's information for terns on what happens to an embryo when it's exposed to a certain level of mercury. And this plot just shows you where there's a higher toxicity threshold that's been established for some birds and a lower toxicity threshold that's been established for other birds. At Egg Island with Caspian terns, the data is just showing us that in the earlier years, one egg out of the 10 that was collected from that site was over this lower toxicity threshold. In 2011, there were 2 of the 10 eggs over this lower toxicity threshold, and then in 2012, 6 of the 10 eggs that were collected were over this toxicity threshold. This suggests, while we're not saying that there is sort of significant population level impacts on the individual birds from this the mercury exposure of the embryos, it certainly warrants keeping a watch on these particular colonies to see if that trend continues. And also, so we know what's going on in the north, and the mercury picture in this area is complex. We have a lot of work to do to figure it out. If we compare mercury levels in herring gull eggs collected across Canada, certainly those birds that are nesting in the north have more mercury in their eggs. So in this plot, we're just looking at the Atlantic coast collections, the Great Lakes collections. I showed you all of those Great Lakes sites where we're getting eggs from. And then there's sites in the Arctic, sites in Great Slave Lake, and then Egg Island. And so obviously the Egg Island sites are up there with the other high Arctic sites, and they certainly, again, this suggests that we should be keeping an eye on the birds that are nesting up there. So in summary, eggs were collected, the earlier ones in June 2009, and not talking about the 1977 eggs, and then from 2011 to 2014. Mercury level, as I said, in the California gull eggs in western Lake Athabasca at Egg Island were are higher now than they were in the 1970s. There's certainly interannual variability in egg mercury levels, and which sort of, right now we can't say too much about trends. We need to do a lot more work in trying to understand what's controlling the mercury levels. This is all methyl mercury, by the way, so we need to know why there's these amounts of methyl mercury in these eggs in these locations. We're saying there's currently no significant risk to the health of the embryos from their exposure to mercury, but because of those data, they're suggesting that some of the tern eggs are above that lower toxicity threshold, that we certainly need to keep our eyes on that. And finally, we need to do longer-term collection of data. Certainly, we need to be up there, and you can see in the Great Lakes, and there's an example here with the herring gulls, temporal trends in the Great Lakes. After decades of collection of eggs, looking at the annual variability and in the levels of contaminants in the eggs, after decades of collection, you can start to put a good trend line on what's going on, but it does take quite a while. So I think it warrants, again, more collections from that area. Real quickly, we got some data recently, so I just wanted to show it as well. Excuse me. On the gull and tern eggs in the Peace Athabasca Delta, this is just the levels of mercury and by number of eggs. So most of the eggs are in the 0.5 to 1 ppm range. We got some American coot eggs recently from somebody that gave them to us, and they're from the near Ward Buffalo National Park. And so we put them in our mercury analyzer as well, just for comparison purposes. These are very different birds, and they're very, very low. So it just suggests that, one of the reasons we're using gulls and terns is that they're the ones that are more likely to accumulate mercury, and we're not seeing that same thing in these other very different birds. Besides trying to do some long-term monitoring, expanding the coverage is one thing. So within a Herculean effort, Dr. Hebert got more colonies sampled in 2014. We're still looking at those data. We're also looking at fish because we want to see if there's any relationship between levels in the fish in where the birds are nesting. Excuse me, these are collected by community-based monitoring program people, and looking at trophic relationships between mercury in the fish and in the birds. And finally, I got to wrap up. I'm going to expire here. Thanks to everybody. It's a huge effort with a lot of people, and a lot of First Nations people help out with this. It's absolutely crucial to the collections and crucial to the program. So thank you.
All right, we do have a minute or two before the break for any questions. I think we have one over here.
I hope I won't have you expire after I have to continue listening there. But one of the critical things with mercury, of course, and there's a lot of interest in Canada in this generally, and certainly even more in the United States, I think, is that it might be, a lot of this mercury might be coming long-range transport from Asia, particularly probably from China, but other countries too. And is there any way that you can sort out that which might be coming in from that long-distance transportation versus what's generated from other places?
Yeah, and that was the purpose for that site in Calgary, is basically because if we saw the same trend in the Calgary birds at Langdon Reservoir, and basically because we didn't, it's a good indication that what's going on with the mercury in the northern part of the province is not long-range transport. There's something else happening up there. And it's basically, and that was the main purpose, to do that big separation from north and south. Global transport's probably coming in to Calgary and into the north, but there's an additional thing going on. And it would appear that for some of the work that's done on to reduce mercury emissions, that we're tracking that in those southern birds because it's going down there. Thanks.
Thanks.
On this way.
Hi, Ron.
You mentioned that you'd measured stable isotopes in some of the golden tern eggs. Presumably that's to assess whether there are differences in where these guys are feeding in the food chain. What did you find?
Yes.
What did you find?
Oh, the increases in mercury cannot be related back to any changes in feeding or food sources. So basically there hasn't been any change, that they're still in the same position. Yep.
Ted Nason with the Mirror. Bruce, you showed some data from the north in general.
Yep.
For gull eggs, and it looked like the values that you're recording at Egg Island and Maui Lake fell into that general bandwidth.
Yeah.
And I was wondering, did you have data from the other northern locations back in time as you do at Egg Island? I think at Egg Island you had 1977.
1977.
Right?
Yeah.
Do you have observations from that
Not
area elsewhere in the north?
Not going so far back with the other ones. That was just happened to be Chip Weslow up there at that time. The other northern monitoring programs, so we do some monitoring in the north for NCP, as I mentioned, Northern Contaminants Program, and for Chemicals Management Plan, which is another federal initiative. And those are much more recent programs, so we're not going to be able to go back that far.
Okay.
Yeah, and to be honest, when Craig was looking into our tissue bank, he tried to get everything that would be useful as sort of old, ancient samples. Yeah, okay. So I don't think those are available.
So we're looking at comparison of contemporary data.
Those are all the last 5 years. Yeah.
So I just kind of leave it. I think it's kind of an open question as to whether there are local effects or not. I don't think we're positioned yet to say.
Yeah. Well, again, this is methyl, and is there something going on in terms of methylation and stuff like that? That's one of the reasons we have to figure out what's going on in that pad region.
Right.
Yeah.
Thanks.
Okay.
Okay.
I think there was another gentleman just sitting down, then yourself. We'll take the next two.
Just a quick question. We know the first fire and bringing a lot of mercury to the local, and I noticed that 2011 it started to increase, and 2012 really high. I wonder if it's because of the first fire in May 2011, and also you mentioned the feed earlier. Do we expect a delay in terms of the feed, or in future monitoring sample analysis, do we see those kind of signals?
With respect to the fires, we did do the dioxin and furan analysis in those eggs as well, and that would suggest if the mercury is coming in with fire, right, as a result of the fires, there should also be a signature of the dioxins and the furans in those. Didn't see any of that, so the conclusion there was that this mercury is in those eggs is not the result of that fire. Like the increase in that time was not related back to those big fires. And the second question was, I'm sorry.
The egg you have concentration and earlier question about the feed.
Yes.
You didn't see any feed. I wonder if the feed should have a delay because of the source or exposure compared to the birds.
I think, so I think, well, I guess one way to answer that is that studies have shown that when there's some inputs, it gets into the prey fish, excuse me, gets into the prey fish really quickly. And so there's like maybe a 1 to 2 year lag, and they've done sort of experimental work on that. So it's pretty quick in terms of moving up the trophic chain to the birds.
One more question over there.
Question about the bird data, the egg data that you have over time.
Yep.
Did you do a statistical comparison between the data that you were able to collect from the 1977 versus more recent data?
Yes.
And what were the...
So there's statistically significant differences in the egg mercury levels in the most recent collected eggs and the earliest collected eggs. So that's both from the 2012 to 1977 with Egg Island, California gulls. So that's the comparison. And also when you look at Mamawi, a 2009 versus a 2012 comparison, you'll see statistical significantly different egg mercury concentrations in those eggs. So those stats have been done.
Okay, great.
Yeah.
And then just wondering, as a follow-up, are you trying to integrate or do comparisons with sediment data in mercury and through time and that type of thing?
Well, yeah. I mean, certainly, and there's, yeah. I'm looking at Jane because the mercury story and everything that's going on, for instance, in the Peace Athabasca Delta, we need all that information. We need the snow. We need the river levels. We need everything really to figure it all out, and then to look at these maybe influences on methylation and stuff like that as well. Yeah, no, to get the whole mercury story, and it's something that Environment Canada, a bunch of us are interested in looking at, and trying to come together and bring all the data sets together, and actually then decide what is the best thing to do in terms of looking at mercury in that neck of the woods. Yep.
Great, it's great. Okay, we'll take one more over here.
Last question. I think you might be being a little too simplistic in ruling out long-range transport by just using the Calgary site, because northern hemisphere transport of toxic materials has been going on for decades and decades. And so you could be looking at a buildup of something that has been transported into the region, and now what you're witnessing is the cycling. So I would be careful in the interpretation.
Sure, yeah. I mean, I guess what I would suggest is there's sort of a more than just those data. There's a bit of a weight of evidence to say that what we appear to be seeing is local input. And one of the lines of evidence is the fact that those trends are different between the north and the south. I mean, we look at the NPRI data and stuff like that. There's mercury input, right? So I think the way that you would probably address that is just to bring all of those lines of evidence together to say it would suggest all of those would suggest that there's some local inputs going on.
Okay, so thank you very much.